Dear learners, welcome to APCCE LMS portal. Today we are going to discuss about the plasma membrane. Plasma membrane is a thin membrane covered by all living cells. This is Dr. Y. Savitri, lecturer in zoology, Government College for Men, Autonomous, Kadapa. Learning outcomes of this topic. At the end of this topic, the student is able to learn about the history of plasma membrane, introduction of plasma membrane, chemical composition, and different model proposed by different scientists and various functions that is performed by plasma membrane are the main learning outcomes of this topic. History of plasma membrane. It is the outermost envelope surrounding the cell that separate and give proper shape and support to the cell, and it is also give protection to the cell. The term cell membrane was coined by Kramer and Nageli in the year 1855. Later in the year 1895, N.S. Overton described that these membranes are made up of lipids. Later in the year 1925, Garter and Grendel proposed that this plasma membrane is composed of phospholipid bilayer. And in the year 1931, J.Q. Flower stated that this plasma membrane is called as plasma lemma. This plasma lemma name was described and named it as plasma membrane by D. Robertson. And he is proposed that this plasma membrane is also called as unit membrane because it consisting of protein and lipids in an organized manner. And it is also present in all organisms in a similar structure, so it is called as unit membrane. Coming to the introduction. The plasma membrane or cell membrane is present in almost all eukaryotic and prokaryotic cells, whereas in bacteria and some fungi, it is modified in somewhat hardness structures because of diversifying environmental condition. It is physically separate the cytoplasm from other cellular components, and it also give proper shape and support to the cell. It measures about 8 to 10 nanometers in size, and it is somewhat elastic, living and dynamic, and it is selectively permeable and semi-permeable barrier because some substances are easily entering into the cell, as well as some molecules they need receptors or some energy to enter into the cell. The plasma membrane is also perform various functions, and it controls the entry and exit of molecules and ions, and it generates some concentration variation between the interior and exterior of the cell. Let us see the chemical composition of a plasma membrane. The plasma membrane consisting of various components. Among that, the three major components are lipids, proteins, and carbohydrates. Among lipids, in consisting of phospholipids and cholesterol. Phospholipids are again, they are neutral phospholipids and acid phospholipids. Neutral phospholipids doesn't have any charge on the surface of these phospholipids. Examples are lecithin and cephalin. Whereas acid phospholipids are the acid in the chemical nature, and the examples are phosphatidyl inositol and phosphatidyl glycerol. Whereas coming to the proteins, proteins are present in two forms. They are structural proteins and functional proteins. Structural proteins are, they involve in the structural aspects of plasma membrane. For example, spectrin of RBC and ATPases of mitochondria. The second one is functional proteins. They are involved in various functions which are performed by the plasma membrane. Examples are alkaline phosphatases, RNases, sodium potassium ATPases, and nucleotides. Some of the enzymes, they act as carrier to transport the molecules from the outer cell to the inside. And the third one is carbohydrates. They are present in short chain structures, and these are added to the sugar molecules. Example, oligosaccharides. Structure of plasma membrane. Plasma membrane consisting of phospholipids. It consisting of 75 angstroms in thickness. Various models were proposed by different scientists. Here we are mentioning 4 models. Trilaminar model proposed by D. Robertson. Biomolecular leaflet model proposed by Danieli and Davison. Lattice model proposed by Hillier and Hoffman. Fluid mosaic model of Singer and Nicholson. Let us see one by one. The first one is trilaminar model. It is also called as unit membrane model. are also called as greater membrane model because it presents in all living organisms. This was proposed by D. Robertson. It is consisting of three layers: the outer protein layer, middle lipid layer, and inner protein layer. The outer protein layer consisting of 20 angstroms in thickness, and inner protein layer is also consisting of 20 angstroms. The middle lipid layer consisting of the lipids with hydrophilic heads, whereas tails are hydrophobic tails. And these lipids consisting of 35 angstroms in diameter. It is present in almost all eukaryotic cells and the structure is very similar. So it is called as unit membrane concept or unit membrane model. Biomolecular leaflet model. It was proposed by Hugh Davison and James Danieli in the year 1935. According to the scientists, the proteins and phospholipid bilayers are arranged as sandwich. The membrane consisting of two layers of lipid molecules and they were coated with the protein. Here in this picture we can see very clearly these are the protein molecules which are associated with the lipids. Lipids are present inside the protein molecules. They are incorporated inside the proteins. The hydrophilic tails are attached with the protein molecules and the hydrophobic tails are towards the inner side of the plasma membrane. Lattice model or micellar model. This model was proposed by Wolfer in the year 1941. Later, Hillier and Hoffman scientists they explained the structure of this model. According to this model, the protein is formed as a framework-like structures outside the lipid molecules, whereas the lipid molecules are present inside the mesh of proteins. Here, 6 to 8 lipid molecules are present as a globule-like structures and they are incorporated inside the proteins. It seems to be as a lattice or network-like structure which are associated. The lipids are incorporated in the protein mesh. Fluid mosaic model. It was proposed by Singer and Nicholson in the year 1972. According to this model, proteins are embedded in the lipids and the arrangement of proteins in the lipids seems to be as mosaic in nature. It simplifies with the help of icebergs which are floating in the sea. Similarly, the protein molecules are also floating in between the lipid layer. It is present in all biological membranes and it is considered as a quasified structures because in the lipid molecules, the proteins are arranged in a mosaic manner. This model assumes that there is a continuous of bilayer of phospholipids in which globular proteins are embedded. These globular proteins are again differentiated into two types, extrinsic or peripheral proteins. These are soluble and they can easily disassociate in the liquids. The second one is intrinsic or integral proteins. These are insoluble and they are not easily dissociate in liquid molecules. And these proteins and lipid molecules, they can move according to their axis and they can move vice versa. So this movement of proteins and lipid molecules in the plasma membrane is called as flip-flap movement. Here in this picture we can see these are the lipid molecules arranged in a flat manner and these are the protein molecules which are incorporated inside the lipid layer and some of the molecules are we can see from the outer portion of this lipid sheet. Functions of plasma membrane. Plasma membrane perform various functions. Firstly, it gives shape and support to the cell and it protects from the outer cellular space. And it also forms as cytoplasmic bridges and microvilli-like structures. Cytoplasmic bridges are the structures which are developed from the plasma membrane towards the cytoplasm. These structures are first described in monkey, pig and Drosophila. Later, these structures are also observed in the spermatogenesis process of human beings. And the second one is microvilli. villi-like structures present in plasma membrane is called as microvilli. These are very similar to the structures which are present in the intestinal regions. They are also called as microvilli. These structures are developed through the outfoldings of plasma membrane. It helps in the absorption of substances which are present outside the cell. Similarly, the microvilli which are present in the intestine is also helps in the absorption process. So this is called as absorption capacity. Like that, the plasma membrane can perform various functions. It acts as a semi-permeable membrane for some substances, also act as a selectively permeable for some substances, and it performs osmosis, diffusion or passive transport, active transport, endocytosis, exocytosis. These are the various functions performed by plasma membrane. The first important function is the osmosis, the passive movement of molecules from a region of higher concentration to a region of lower concentration. Similarly, the movement of water molecules from the concentration of low concentration to the high concentration. Here, these molecules like water, carbon dioxide, they can move very easily from outer side to the cell to the inside of the cell. It doesn't need any energy and it doesn't need any ATP-ASS. The osmosis process is depending upon the size of the molecule. Smaller molecules can enter into the cell very easily. The diffusion or passive transport. In this method, the movement of molecules from a higher concentration to lower concentration is transported with the help of transporting proteins which are present inside the plasma membrane. Here the transport of molecules doesn't need any ATP or energy. Here the molecular size is very small and they can easily pass through the receptors which are present inside the plasma membrane. This is also called as facilitated diffusion because here the molecules need the help of a receptor, which maybe the receptor is the protein molecule. The substances which are not soluble in lipids like glucose and amino acids can pass through the plasma membrane with diffusion or passive transport method. Active transport. In this mechanism, the substances move against their concentration gradients. The larger molecules have to move inside the cytoplasm. Because of the largeness of molecules, they require certain receptors inside the plasma membrane. These receptors may be the proteins. And for the movement of molecules from the outside to inside, they require certain energy. This required energy is taken from the mitochondria. This movement is called as uphill movement. It can be studied under three types. The first one is ATP driven active transport. In this mechanism, sodium, potassium, calcium ions are transported from the outer to the inside of the cell. And the second one is the light driven active transport. In this mechanism, the light is driven the transportation of molecules. Under this, we can study the halobacterium, or it is also called as halobium. In this bacteria, the bacteriorhodopsin, that is H plus pump, and halorhodopsin Cl plus pumps are driven by light. The third one is ion gradient active transport system. In this mechanism, amino acids and sugars may be transported in different mechanisms, that is symport, antiport and uniport. In symport, the molecules are transported in single direction with the electrical gradient of ions. In antiport, the molecules are transported against the movement of receptors. In uniport, single molecule can enter into the cells at one time. And the active transport can be explained with two examples. The transportation of glucose and amino acids which is present in the lumen, they are absorbed into the walls of small intestine. And also the filtrate which is present in the nephron, they can reabsorb into the collecting tubule. Endocytosis. Endo mean inside, cytosis mean taking. Endocytosis means the intake of substances from the outer cellular environment to inside the cell environment. It is also called as intake of substances from the extracellular fluid into the matrix. It happens in three types. The first one is pinocytosis. The intake of small droplets of liquid substances is called as pinocytosis. It is occur in all types of cells. The second one is phagocytosis. The intake of large droplets or solid substances into the cell is called as phagocytosis. It happens in specialized cells like neutrophils. And the third one is receptor assisted endocytosis. The intake of specific molecules with the help of specific membrane proteins which are present inside the plasma membrane is called as receptor associated endocytosis. Here the receptor is modified according to the shape of the molecule. Exocytosis, the sending out of unwanted particles from the cytoplasm to the exterior is called as exocytosis. Here the excretory substances which are present inside the cell, they may be sent out through the plasma membrane. Here in this picture we can observe this is the cytoplasm. These are the excretory particles which are present inside the vacuole, and this vacuole is moved towards the plasma membrane, and the substances which are present in this vacuole will be sent out through the plasma membrane. Summary of this topic. Plasma membrane is surrounded by the cell and it gives support and shape to the cell. The name plasma membrane was described by D. Robertson. This model is also called as unit membrane because the unique presence in all types of animal and plant cells. It protects cell from the outer environment and gives support shape to the cell. The plasma membrane is selectively permeable. That means it allows certain molecules which are low molecular weight, and it is semi-permeable. Certain substances like water, they can move easily across the plasma membrane. This is called as semi-permeable. And the structure of plasma membrane was explained in four models. Trilaminar model was proposed by D. Robertson. Biomolecular leaflet model was proposed by Danelli and Delson. Lattice model was proposed by Hillier and Hoffman. Fluid mosaic model was and Nicholson. Among these, the fluid mosaic model who proposed by Singer and Nicholson as the accepted one. All biological membranes consisting of a similar structure, that is trilaminar structure, so it is called as unit membrane model or gated membrane model. It consisting of various some components like proteins, lipids and carbohydrates, and it performs various functions. The extensions of plasma membrane is formed as microvilli and plasmodesmata, and it performs different functions like osmosis, active transport, passive transport, endocytosis and exocytosis. This is about plasma membrane. Thank you.